Great. I'd like to thank the organizers for this opportunity, and thank you for the introduction. My name is Seth Frietze. I'm an associate professor at the University of Vermont, about an hour and a half south of here on beautiful Lake Champlain. The work in my lab, one of our focuses is to understand cancer progression, and that's the initiation and progression of cancer to metastatic disease. And this process really involves some key molecular events that happen where a differentiated cell type loses its differentiation status and can undergo very many molecular events to become metastatic. And so we're really interested in generating primary data that helps us get an insight into the epigenetic processes that control these gene expression programs, including transcription factor activities and histone modifications. And we'd like to incorporate these data sets together to maximize our biological inference. And we do that through some gene regulatory network analysis. And the overall goal of this is to identify important cancer vulnerabilities so that we can further study these as potential cancer therapeutics. So this is largely the work of a graduate student, Kong, in my lab. She couldn't be here today, unfortunately, but her work really relies on a MCF10A normal mammary epithelial cell, a human cell line, and its derivatives that represent a cancer progression model. So this model involves a stable introduction of a transgene, a constitutively active HRAS oncogene that renders this 10A cell line tumorigenic. And then this cell line went through a xenograft model to generate a lung metastatic cancer cell line. And these cell lines largely are genetically similar and represent many of the features of cancer where these mammary phenotypes are preserved in the 10A cells, but the AT1 cells are more proliferative, and the CA1A cells demonstrate many of the molecular features associated with metastatic state. And so with these cell lines, we've managed to generate a number of different ChIP-seq and other functional genomics data sets, including RNA-seq and other modifications. And we're particularly interested in studying the cis regulatory elements that are called enhancers. Then there are a number of different enhancer classes that can be defined by combinations of different histone modifications, including H3K4 monomethyl, which represents a poised enhancer, but that enhancer may, over cancer progression, obtain a H3K27 lysine acetylation mark, and then that element can then obtain more K27 acetyl to be called a so-called super enhancer. So this is some of the underlying epigenetic program involved with these gene expression changes during cancer progression. And we'd like to integrate some of this data and perform gene regulatory network inference to understand some of the transcription factors and features that are changed in this progression model. And so one way to sort of integrate all this data is to perform chromatin state annotation with chrome-HMM, which takes these histone marks and is able to look at their different patterns and provide an annotation of your genome from MCF10A cells into different functional states. And so shown here are some of the signals of these modifications and a 20-state model that generated these annotations, including some repressive heterochromatin domains, as well as enhancer or gene body type of information. We also included histone H4, lysine 5, and lysine 8 acetylation marks, which represent euchromatin modifications. We noticed that by including this mark as well as RNA, we were able to identify subsets of enhancers that either were active by having K27 acetyl and K4 monomethyl that had low H4 lysine acetylation, or enhancer regions that had high H4 lysine acetyl regions. And so we're very interested in studying these. And so this alluvial plot did not come out. So this is supposed to be an alluvial plot that then generates the transitions from one state to the next. So what I'm showing here is the MCF10A enhancer states that are colored here by, say, for example, these poised or bivalent enhancers that are classified in MCF10A cells. And what's not showing up is the conservation of these states as we look across the breast cancer progression series. Oops, sorry. And what this should show is how dynamic these states can change from one cell line to the next, indicating that there's a change in the enhancer calls across breast cancer progression. And we're particularly interested in. I just wanted to show, so these chromatin states is a great way to pattern the genome, but it's important to look at your signal and especially to validate some of these findings and study it, because it's one thing to stick your data into a model, but it's another thing to believe your signal. And so a talented bioinformatician in my lab, Joe Boyd, who has a poster on this, has developed a R package that uses reduced dimensionality to represent combinations of functional genomic data enrichment patterns across the genome. And what I'm showing here is a t-SNE plot that, this computer is a t-SNE plot that is generated by clustering features composed of bed files of regions of the genome. And here we're looking at active enhancers with histone H4 lysine acetylation. And the clustering of these patterns, these features are driven by the signal of multiple marks. And what we can see when we look at this, this tool allows us to explore and interact with the data. And we're currently developing it into a Shiny app to be able to use this to represent a reduced epigenome and study many different modifications at the same time. What we can see is by comparing MCF10A to AT1, the tumorigenic cells, there's patterns of these features, of these enhancers that gain or lose specific modifications over time through the breast cancer progression series. And highlighting here in the red box are an area of active enhancers that are high with histone H4 lysine 5 and 8 acetylation in MCF10As, but lose that mark as 10A AT1 progresses. And so we're actively studying these features and their functional significance with breast cancer progression and gene expression. Another area of interest is to identify super enhancers that are highly active enhancers linked to lineage specification. We used rows, ranked order of super enhancers to identify large super enhancer domains, and binary clustering shows that each of these cell lines has a unique distribution of super enhancers present in 10A, AT1, or metastatic cell lines. And these features, these super enhancer features, correspond to more transcriptionally active regions in 10A, for example, or in CA1A, the higher expression of genes associated with these super enhancers. Moreover, these genes associated with these cell line specific super enhancer domains have annotations or ontologies that make sense with some of the biology we know about these cell lines. So in order to incorporate this data, there's a number of tools that are been generated to integrate gene expression and chromatin data. And one of these is the so-called CRC mapper that is used to generate core regulatory circuits of cells. And this tool, we used bulk RNA-seq data corresponding to the different cell lines, as well as our H3K27 acetyl chip seq data. And ultimately, the goal is to identify master or critical transcription factors in each cell line. And this involves identifying auto-regulated transcription factors associated with super enhancers. And the output of this is a target list of active transcription factors present in the MCF10A, AT1, or CA1A cell lines. And among these are interesting transcription factors that could play an important role in the regulatory circuitry of normal or cancerous cells. We also performed integrative analysis with PICA, which is a paired gene expression and chromatin accessibility to map gene regulatory networks. And I'm showing here the global net. I'm sorry, this is a very sensitive computer. I'm showing the global network here, highlighting some of the top transcription factors in this network. And we can visualize the transcription factor activity by this dot plot showing both its stress score in the network as well as its expression levels. And this PICA analysis provides a statistical comparison, a differential network analysis that can highlight key changes in transcription factors that are different between different cell states. And so by ranking these transcription factor degree changes between 10A and AT1, for example, shown here, or statistical representation, we can map these networks and identify transcription factors that are more active in these gene regulatory networks in different cell states. And so just to highlight the importance of some of these integrative analysis, we're able to use these to find key transcription factors such as RUNX1, which is a known tumor suppressor in breast cancer. In the cell lines, why is this going by itself? This computer has a mind of its own. In cell lines, I think it's my slides probably. In these cell lines, RUNX1 protein expression is high in 10A and it goes down in AT1. And this data is reflective of a concatenant change in epithelial to mesenchymal transitions. I'm going to stop. All right. And then this data in TCGA data, you can see RUNX1 expression is high in both early stage and decreased in late stage lung cancer, or breast cancer, excuse me. And importantly, the expression of RUNX1, high expression, is a more favorable outcome in breast cancer patients. RUNX1 is one of the top mutated genes in breast cancer, and those mutations are often found in the DNA binding domain or a domain that interacts with its obligate partner, CBF-beta. And so we're currently studying RUNX1 as a tumor suppressor, and we've established Dagron cell lines to acutely deplete MCF10A and study the effect of phenotype and epigenome that RUNX1 plays, as well as other oncogenic factors, and studying their role in these gene regulation. So in summary, we performed epigenomic mapping on this 10A series and used chromatin state annotation to identify dynamic regulatory elements. We've incorporated some new tools in this process that allow us to integrate and visualize this data. We've taken advantage of two gene regulatory network tools to better integrate this data and understand key transcription factors in this process. And these results highlight important tumor suppressor or oncogenic transcription factors in this cell model. Finally, I just want to thank my group, including Joe and Julius, who are here, who both have posters. So please go check them out at this lunch break in a minute. And I'll take any questions.
Maybe I should ask one. So the tools you showed, are they available?
That's right. So are you talking about the chip T-SNE? Yeah, so that's an available package that we're right now on GitHub. It's in development, but it is the developers here, and and we find it's going to be a very useful tool to visualize multiple combinations of marks. So yes, it's a R package that uses BAM files or bigwig files.